yeah, you found that interesting what I was telling you this morning. I was not. It's unusual for me to give a lecture like this, and I was surprised by some of my own slides, and I could not see the next slide in this mode that I had on because I had pictures of the people. So I never knew what was going to be the next slide or the next build in the presentation. What we usually do here is that you get embryos, and you have to sort the embryos into different stages, and then you have to find mutants among these. We cannot do this now. It's unfortunate, because what I learned is that in the zebrafish part, what is nice is that you actually do something with an organism, that you see embryos, that you actually do something practical, even if it's as simple as just taking the embryos out of the shell, out of the chorion. So the chorion is the eggshell, basically. And for the most part, this is easy listening, I think. It's not really complex. There's no formulas. There is no... It's very evocative what we do, because seeing is believing is part of this. It's just we look at what happens, what cells are doing, how an embryo develops. And I must say, for me, when I was a grad student, I did mouse development, but nobody was able to explain actually how this embryo is forming. There is this thing about, there's primitive streak, and then you end up with a chick embryo, but the chick embryo is not the mouse embryo, because there was nobody able to explain any of this, what's happening. And I must say, embryonic morphogenesis, I only understood how this is happening when I looked at the zebrafish. So when I started postdoc at zebrafish, that was the first time that I really understood, okay, this is how an embryo is forming, and this is actually the power of the system, that we can look and watch things as they happen. It doesn't seem intellectually challenging at first, but it's a different way of thinking. We don't think about molecular mechanisms here, we think more about cellular mechanisms. What are cells actually doing? And this is different from looking at expression levels, doing single cell sequencing and things like that. It doesn't mean it's not intellectually demanding. It is actually, there's a lot of room for creativity here. It's a matter of, and this is what this is about, is trying to describe in words what you see. This is a bit like an artist. And I've seen people, postdocs who are not able to really describe, put into words an expression pattern and these things. So I think this is what we try here to do, is now, just we look at these videos, we look at these drawings, and try to really just describe what we see without talking about molecules and anything like that. And I think this is really helping us to sort of conceptually understand what's going on the morphogenetic level. And then we can try to understand how this is done on a molecular level. But when you talk about molecules, it's usually about regulation. And I must say, when you hear the news, we say something regulates is the same thing as doing something. We say Hox genes make antenna, Hox genes make wings, Hox genes make legs. Well, that's not true. They just regulate something. They don't do anything really. So when we talk about regulation, we haven't actually understood anything, how this is done. Something is regulated. It's like turning the switch on a heater, but the switch on the heater doesn't make the heat, right? So what we do when we look at cell biology, when we do morphogenesis, we actually look at things that are happening, and maybe even the forces, how forces are transmitted, how cells are attaching to each other in a much more sort of mechanistic way. Rather than regulation, we actually want to understand what are the mechanics of the things that are happening. And so what I have opened up now, I have opened up on my computer just the first folder. I think it's called Cleavage Stages. And there is 6 videos in this folder. And And then on the same side, you can have open this part A drawings, okay? And so what we try to give you the feeling here is you imagine yourself, virtual reality, that you sit at the microscope, you have this embryo under the microscope, and this is exactly what John Min has done. He had these embryos on the microscope. He actually had the microscope at home because of quarantine. He didn't feel like going to the lab and going back and forth, so he just took the fish with him, the embryos and the microscope, and used his iPhone, his cell phone, to make these videos just on the microscope. So you could do this yourself. You could have done this also in the lab, probably under normal circumstances. So I propose that I share my screen now, and you know, we just talk through this, right? So that we are round off. You know, we are about
maybe it's better if
dozen
one of the
people.
students
What?
share.
Yeah.
I think that maybe it's better if one of the students is taking one movie, share a screen, and try to describe it. So
we can do that too. Yeah.
And we can talk over if.
Yes.
That,
Yes.
I mean, is that okay for you, or you prefer to just listen? I mean, at least you do something.
Yeah. Actually. Yeah.
I think maybe it's easier if just if just one person actually streams his desktop.
Yeah.
Because that way we don't have to switch all the time, but we can still just describe it to students. I mean,
yeah, we can have my because I want to point at things with my mouse, and we see how it goes. Okay. I mean, there's no pressure here. I mean, we all, you know, we try to engage you as much as we can. So I start sharing my screen, and then I go.
Still working. Okay.
Yeah. Okay, you can do this also on your monitor if you want to. And then I played it a couple of times. You can play it a couple of times. So this embryo is, I think it's about I'm not sure, half a millimeter in size, diameter. Maybe a little less, okay? Yeah, so this is the X that you get when you come to the lab, and then you see this is what it looks like. So Jiangmin takes the needle and moves it around a little bit, but basically you see everything that you need to see when you look on the side. Okay. Let me pause it here. Basically, you see everything that you need to see here. So what do you see? Everybody can say. It's obvious, but how to put it in words, what you see? It's just somebody start. Okay, I ask specific questions. Yeah? 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 Yeah, that's the chorion. No, this is real. This is real. That's why we turn it around and you see. Yeah? Right. Right. Yeah. Yeah. Right. So what is the bulkier part?
The egg yolk.
Right, that's the egg yolk.
Exactly.
And what is on top?
The embryo forming.
Right.
Well,
I mean, it's all
or-
the embryo.
Yeah, but
There's a separation in the embryo into a yolky part and into what we call the cytoplasmic part. Okay. We don't have that here. If the embryos are laid, if the eggs are laid and just fertilized, this thing is not there. Okay. It's all round, and then there's the separation going on, and that's all you see basically. You see a yolky part and a clear part, and I'm just telling you now. And what you see then, of course, so I can somehow, how do I do this?
What do you want to do?
I want to switch. I mean, I must have a, yeah. I switch to this one here. You see that? You see the one cell drawing? Okay. Okay, so you've pointed out this clear part here, right? And then, so that is what we call this, basically the cell, that is the cytoplasm. And this is the yolky part. If you look at this, you see a couple more structures here, or strange things. In the yolk, you see stuff, right? No? I mean, if you look here, you see this? You don't have the... Yeah, there are little groups in there. Yeah. And that happens, and that is part of that process of separation of, because this is called cytoplasmic streams, okay? This is, you have to imagine the separation occurs by liquid moving from the yolk into the cell. That's how it inflates. The cells becomes inflated. And these are these streams that you see here. And then you see this brown stuff here. And this is just some granular material that is accumulating at the interface between the cell and the yolk. Okay. Yeah. That's all there is to say. So they drew that here. So here it's a bit, it looks like granular stuff here. But basically, and here's these cytoplasmic streams. So the cytoplasmic streams, they're not just liquid, they also contain protein and RNA. So there's a transport of RNAs and of proteins from the yolk into the cytoplasm. At this point, I'm not exactly sure, you still may have two nuclei here that you don't see. And this is the time when you have, what do you call this? I forgot, nuclear something. So you have a male and female nucleus, paternal, maternal, and you have it.
Zygotic forming.
It's a zygote forming, yeah. There's a term for that, I forgot. Anyway, karyogamy. Yeah, okay. Anyway, Next movie, 2-cell stage.
You have to, okay.
Yeah, no, it takes a
Yeah.
while.
Yeah.
Okay, you have it also on your desktop, or you watch it. Is it okay just to watch it on the desktop, on the?
Okay.
Well, this is easy. Just somebody say something what you see here. I mean, it's basically the title, but
On the granular phase, you have these 2 cells.
you have 2 cells, yeah? That's right. And how would you describe? How would you? And this is important now. So you have 2 cells, so there has been a cell division, right? How did the cell division occur? So cell division means the 2 cells separate, right?
Mm-hmm.
But did they separate completely?
No, they're still in touch. So they kind of open at the bottom where the granular, they are
Right.
connected in the granular phase, kind of.
Right. Right. So yeah, that's the whole point. It's an incomplete cell division. It's called superficial cleavage divisions. Okay, they're superficial, and that is different from other systems. It's different from frogs. In frogs, the cell division is complete. So actually, the first cell divisions, they subdivide the yolk. So we have sort of an embryonic disc that is forming. You'll see that later. And the cleavage plane just goes down to the yolk, but they stay connected to each other and to the yolk. So in principle, they're still syncytial. So I'm doing this. Okay. So yeah, not much had happened, but you have now these 2 cells, and the cell division has been here. Okay. And maybe something you notice is that this embryo now is 45 minutes old. Okay? Yeah. Yes? Mm-hmm. No, they're 2 cells. They're considered 2 cells. So I'm not an expert on mitosis, but I would think that cytokinesis occurs normally. It's just that the cell membrane is not pulled in all the way. But yeah, okay. Next movie. Oops. What is the next movie?
So
1 hour, right? Yeah.
yeah, it's 1 hour.
So 25 minutes later, we are here. I must say these embryos are a little bit dirty. So you see this stuff on the side. That's mold that's trying to attack the embryo. These little threads that you see. That's okay. It's not such a. You don't see much. but it's clear it's 4 cells.
So what else you can say about these 4 cells?
It's a real question, right?
Yeah.
I don't have an answer.
No, you. I'm asking them.
But I don't know what you say. I don't.
If you see the orientation of these cells, how are they?
Oops.
Yeah, you're already at 8.
Oh,
This.
okay.
So
What happened here? So when you have 2 cells, it's like this, right? From the top view. And this would be the embryo around it. So the question is, Alessandra, which orientation is the cell division?
Yes, exactly. So what can you say? So is I mean I cannot put it on your screen, but is it something that makes 4 cells dividing 2 bottom to
So
top?
So it
or
could be, in principle, it could be this way,
exactly,
or it
or
could be this way.
exactly. So that's
a question to the students.
yes.
Do you have a situation where the 2 cells are divided like this to make 4 cells,
So you have
or the other way, where you would have, oops, that doesn't work. Or would it?
Just make another 2 cells and divide. Yeah,
Yeah, it's not so. I'm not an artist. Or is it?
Uh,
This way? Well, just is it A or B?
It's B.
okay.
Who is for B? Yeah. Who's for A? Everybody's for B. I'm also for B. Okay.
Does this direction of dividing have a name, or?
I have no idea. So these are all cleavage divisions, and that's, and I think, well, I have never thought about it. I take it for granted somehow. I mean, it's some things you don't care about, and I haven't thought about it. They are perpendicular. I think there is. I think when I was first semester, we talked about this different cleavage patterns in different organisms. So there are certain cleavage patterns in sea urchins and in certain snails, but and they have all terminology, but that's, I don't know, that's 25 years ago, so I don't remember. Okay.
And are they completely symmetric, or do
Yes.
the 4 cells differ?
They are completely equivalent. So we move to movie number 8 cell. So it's already a little difficult to count, but if you imagine I think you get the idea. and these are simply 8 cells. And if we look at the drawing, it's here, you see 8 cells, and if I draw it for you now, it would be, can I duplicate that? No. You know, it would be sort of like this, 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 and this. So then it starts looking a bit more rectangular. Yeah. So if you then compare, you had this situation here of having 4 cells. So now you divide these things up, and then you have 2 new cleavage planes, and that's how you get from 4 to 8. Okay. This looks like a Tessiner Brot, right? Okay. What is important is we saw 1 embryo, 1 cell, 2 cell, 4 cell, 8 cell, and then we can go to 16 cell. Actually, this is all the same. See, this is just more cell divisions. It's actually always looking quite nice here. So you see 1, 2, 3. Suddenly you have more rows of cells. So that is characteristic. So when you look at, and I go back to the drawing now. Where's that drawing? Here. The main thing that happens here is, you know, if you think about this is a single cell, and here, you know, yeah, let's say 32 cells, it doesn't matter. What is the difference between the cells, between a 2 cells cell and a 32 cell? What is the main difference when you think about cell number and cytoplasm?
The size.
Yeah.
It seems to be smaller.
Right. So the cells are getting increasingly smaller. In cleavage stages, there is no cell growth. There's a given amount of cytoplasm, and that's continuously subdivided. So the ratio of nuclear volume to cytoplasm is actually increasing. Okay. Yeah, you can always ask. Yeah. There's not much going on. There is translation, and there is a lot of cell division going on. I mean, a lot of DNA synthesis. There is no G2 phase. There is no interphase. I mean, if you imagine, you see that later, you know, they just keep dividing, All the material is there, most of the protein is there. There is not much going on. It's just producing material. In terms of number. Again? No, not that either. They're all equivalent. You cannot differentiate anything. What you have is you have the animal side and the vegetal side. So you do have an axis. The animal side is where the cells are, and the bottom part, the vegetal part, is where the yolk is. So you have that body axis. So the embryo is basically radial symmetric, and it stays like that for quite a while. What do you see now? You see an embryo, right? The movie? Nope?
No.
Okay. Here we go. So this is the 32 to 64. Actually, it's a 64 cell embryo. And I don't want to torture you too much. I mean, you can't distinguish cells anymore, right? That is the basic, that's the basic message here. And going back to, so basically it's this situation here. So what is new here now? This is now something really new. Compared to especially the early stages of cleavage, when the membrane was not completely pulled in.
And the cells are not in one plane, but
Exactly.
Right.
right on top of each other.
Right. That means you had this. This is a new plane of cell division here, right? You have now a population of cells that's on the outside and the cell that is next to the yolk, right? And the cells are completely cut off from the yolk. Okay? This has practical consequences for us when we do research. If we inject in this stage, also up to this stage, if you inject a substance, let's say in short RNA, an iRNA, or a morpholino, or an RNA, or a DNA into this embryo here, it will be taken up in all cells. Not in the same level necessarily, because there's always stochastic differences. If you inject here, there will be more stuff going here than there and stuff. But if you put it, let's say, we usually try to inject into this region here when we do injections. And then this is where the yolk is really less dense. And then you get very good distribution of these substances in all the cells. But in this stage, you cannot do that anymore. What happens in the yolk stays in the yolk.
Also for chemicals.
No. Well,
okay. They still go by diffusion or transport or yeah.
If you have these small molecules that people use,
yeah.
They go past the membrane. They are cell permeable, right?
Mm-hmm.
Things that are not cell permeable, they don't go through anymore.
So there is not active transport of impermeable stuff through the cells anymore from the yolk.
I would know about that.
Oh, okay.
But this is now a clear separation between the yolk and the embryo. You're right in some ways. Maybe we see that later. we go back to the video because we have a lot more things to discuss. I mean, we are moving a little slow here. Oh, but we are just done with this one. Okay, now I stop the sharing here. Oops. I will restart the sharing in a second.
Now we go to the next stage, no? To the...
We go to the next folder. Yeah. And we will skip a few stages, I think. I do the sharing again, start sharing. And you see that?
Mm-hmm.
Okay. I will go to this here. By the way, this is a 1,000-cell embryo. Why can we say this is a 1,000-cell embryo? Well, you can count the cells. It is also just an estimate. How many cell divisions does it take from 1 cell to 1,000 cells?
Oh.
Roughly. I mean, 1,024. I'm waiting for an answer here. I mean, you can do this in your head, or you can write it down.
10 divisions.
10
cell divisions, okay. So now this embryo is, how old is this embryo?
3.5 hours.
So it took 3 hours for 10 cell divisions. I mean, one thing is, we call it 1K, because it's about 1,000, and that means cell divisions have been synchronous. So there have been 10 synchronous cell division. I mean, this is this thing also about the coronavirus, right? You have these doubling times. And it's amazing that you get just 1 cell and then 10 doublings, you had already at 1,000 cases, right? Has anybody done any cell culture, HeLa cells or COS cells? Do you have anything? Have you no practical experience with cell culture? Have you? But just then, how do you think when you take a cell in cell culture? How often do they divide? How fast? What is the division time?
Roughly, yeah.
Yeah.
Mm-hmm.
Under optimal conditions, right? Right. And a normal cell culture cell is usually about roughly 18 hours. Yeah, 18 hours. So I used to say 1 division per day, right?
Yeah.
A bit faster, but it's 1 per day. And that's a mammalian cell under, with good serum and all that. And this is now, it's like E. coli. It's, it's 3 hours, 10 divisions. It's on average, it's 30 minutes for cell division. So that's really fast. And so that shows there is no time for interphase. There's no G2 phase. It's going from mitosis basically back to mitosis. S phase, mitosis, S phase, mitosis. Okay. Then I just show the video here. And then cell cycles slow down. You don't know that, but I'm telling you this. And then this is a nice face. It's called oblong, but I think this is also the sphere stage. This is actually
not working.
No?
No.
Okay, sharing is paused. Bring your shared window to the front. Resume share.
No, nothing happens.
Then I do like this again. Okay, so.
Okay.
So this is, I don't know how many cells there are. I guess probably 3,000 or so. No idea. But it's very clear to see that, you know, these cells have become sort of, it looks all granular. These are little small cells. This is actually when people do cell transplantations from embryo to embryo, this is basically the stage when they like to do that. The cells are just loose. They are contained under a membrane and they're just loose under it, and you just can suck them out and stuff them into another embryo. So this is a great stage to do that. Yes? Yeah. Yeah. This is before the dome stage. Well, I don't quote me on the 3,000 cells, please. Yeah, dome is afterwards. Yeah. Can you see this now? No.
No.
So I cannot switch easily the videos. Okay, so then I have to stop share and share. So this doesn't work. Okay, so okay, so now
Okay, now we can see.
Tell me what you see here. Ah, the stupid pipette there, but you get the idea. Okay, so I go to the drawings and then you can tell me with the drawings. So from here to here, there's a first change, and I think you see it. So what has happened here?
Either the yolk grows inside the animal cells or the animal cells grow outside the yolk.
Yeah, yeah. Well, it looks like the embryo is buckling, right? It makes this, it gets this indentation. So this is actually the beginning of end of blastula, beginning of gastrulation. The cells are starting to become active. They're not just dividing anymore, they start doing things. So this is actually the beginning of what we would call morphogenesis. Morphogenesis means acquisition of shape, right? And so you have a shape change in the embryo here. And I'm not an expert particularly on this part, but I think there's a lot of cell division going here and here, not cell division, cell movements. The cells start actively moving and they move to the sides, like here. And then somehow the yolk is is probably under pressure, and then there's some loosening here, and the embryo is a combination of cells moving out and the yolk pushing up.
Also, the cells around that are touching the yolk are starting to flattening, so they're also changing the shape of each cells. So then also the one at the margin, and you know, that.
Yeah, which is something you don't see here at this resolution, of course. Okay, now we move to another folder.
See?
Which is
The shield, or
hold on a second. No, 50%. Why don't I?
yeah, 50. The first one is the 50% apably, and then the shield stage.
Yeah. Okay. Okay. I have to hold on a second. Sorry about this. 50% is where? Oh, I don't have the, yeah, here. Okay. I know this didn't work so far. I have to do this again. Okay. Okay. So this is a bit more of the same, but now you have a clearly, you see that this embryo, how would you describe the shape of the embryo? Just one word.
Like a bowl.
Like a?
Like a bowl.
Like a bowl, yeah. Yeah. Or like a cap or like a cup. Like inverted, of course, right? It's forming a cap on the yolk. But if you would look at this now from the what we call the animal view, which would be the top of the embryo, you would be not able to see anything remarkable here. Okay. And this is at 5 hours. And what you also see is this bowl is, you know, how much does it cover actually of the yolk, the embryo? In percentage. I mean, it's already in the name, but just say it. I would say it's 40%, 45. It's not even 50 yet. It hasn't quite reached this particular embryo, the equator, right? So epipupillary stages it's a cell movement over the yolk. And we say 30% epipupillary, 40, 50, 60, 70, 80. And at 50%, epipupillary, that's what we call it when it's basically at the equator of the yolk. That's half the yolk is covered by cells. And I go quickly here. So that's basically like this. And if you would now look from this perspective here, it would be a circle. And it would just look like that. Okay, there would be a bit, it would look a little denser on the outside than on the inside, but that's because if you look at it, there's more cells here than if you compare here, because there's more density. If you look at this, there's more cells here that you look through than you have to look through here, right? So that makes it appear a little denser here. Okay, now, and this is an important stage. And I share this now with you. Now this is different. What do you see here? What is different now? Anyone?
And the edges are thicker than the other parts.
There's something here, yeah. Oops. There's something here, right? And also something here. So this is thickening, what we call at the margin. Margin means the edge of the embryo. It's called the embryonic margin. So this is a margin, and here's a margin. The margin goes all around, but there is a differentiation on this side and on that side. Maybe you can, I play this movie again. Maybe it becomes more obvious. So here it's very thick. Here it's also thick, but not quite as thick. Yeah, this could be done. This video, we make a new one for next year. This is not so perfect. Nevertheless, this is called the shield stage. So what is this shield stage? That is shown here. So this is what you saw. This was this thickening here. And if you look from the animal side, right, this is animal view is from this side. And that is shown here. That's the animal side. So if you look from the top of the embryo, you see there's this structure here, and that is called the embryonic shield. I will give a lecture after lunch, and I will talk a little bit what the shield is, what it's about. But clearly there's more cells here. But we don't know why there's more cells here. Is it more cell proliferation? Is it cell migration? But this thing here is also at the margin. So the margin is all the way here. And there would be also this thickening is going all around here. Just more here and here. And then you can see it in the movie, there's a fold here. So we don't know, is there something happening? Is there something folding in? Is there cell migration here going in? Or is like a tissue invaginating? We don't know. But this is, by some people call this the onset of gastrulation, because gastrulation is the formation of the germ layers. And you all had some developmental biology, I think, with Marcus. You heard his lecture. So you have ectoderm, endoderm, mesoderm. I'll talk about this later in the lecture. Today we just see, we discuss what we can see, okay? We don't have to think about mechanisms really at this point. And hold on a second. Gotta get better at. So now I move to the video 90% epibuli, okay? I skip one. Okay. This is 90% epibuli. Now there's two things that you can see that are important about the symmetry of the embryo. So we had the shield. And we have the yolk. So I can tell you, so this is epiboly. So this would be, if I can get that needle out of the way, that bugs me. Let's say, let's do it again. I wanna, oh, no. Okay, so you know what this is? That's the leftover of the yolk, basically. That's called the yolk plug. It's the last piece. It's the last piece of yolk that is not covered. That's why it's not 100% epiboly, but 90% epiboly. Now, what else do you see here? If you look at the amount of cells covering different parts of the embryo, is it symmetric or is it asymmetric, or how are the cells distributed over the yolk? Do you see, I mean, do you think anything that is... Okay, I ask you then more leading questions. Is it uniform, the cell distribution? Yes or no?
No.
No, okay. So where are the cells? Are they here or are they here?
There are more on the bottom left.
Yes.
But it seems
So,
like
so they seem to be all on this side here. So somehow we had the distribution of cells has changed from being symmetric, radially symmetric at 50% epiboly. Then we had the shield, which you will learn later. That's the first sort of anatomical or visual hallmark of a dorsoventral pattern. And then you have all cells on one side rather than on the other side. And that you can see again here. So we have something remarkable has happened. So here, the embryo is radially symmetric. You have an animal pole and you have a vegetable pole, okay? Now, and this is a giveaway for later, you have a differentiation. Suddenly we have a differentiation along another axis now, and that is this axis here. Because this side is different from that side. And just so you know, this is the future dorsal side of the embryo, and this is the ventral side of the embryo. So here and here, right? So that's dorsal, that's ventral, that is ventral, that's dorsal, and this is dorsal, big time dorsal, okay? At shield stage. And now we saw something like this now, okay? We saw it the other way around. So I could rotate this and flip it, but this is basically it. And just, you cannot know this, but at shield stage, this is where the shield was, okay? So this was dorsal. So this is the back of the embryo. This is the ventral part of the embryo. And what have we found now? What has happened now? And this is now everyone's guess. What do you see now here? What you know, you have now two axes. You have an animal vegetable We have a dorsoventral axis. So if you look at this embryo, where would you say, where's the front of the embryo? Is it at 12 o'clock, at 3 o'clock, 6 o'clock, or at 9 o'clock?
I would assume that at 12 o'clock it's the head, and towards 6 o'clock in the bottom it's going to be the tail.
Right. So actually, this is exactly what has happened now. We have now formed the body axis. Okay. There's a few cells here. They matter a little bit later, but they don't concern us at the moment. But basically what has happened from here, starting here, we have a secondary body axis, and these axes together, the animal vegetal axis and dorsoventral axis, basically forming here already the anterior posterior axis here, A to P, which means the animal pole becomes the front of the embryo and the vegetal pole the back of the embryo. What you have to consider, though, is that we have ectoderm and we have mesoderm or mesendoderm. Just to make it simple, we call it just, it's mesendoderm, okay? But for the most part, endoderm, it doesn't concern us so much. So this layer here, that's the mesoderm. So there's some complex stuff going on. We have cells here, and they're not quite at the front. Who knows who becomes what tissue in the end? That's what I will discuss in the lecture, okay? But basically, we have in terms of morphogenesis, we start getting a little bit of a grip. It would be best, of course, if we would have a time-lapse movie. Then we could really follow all these steps and follow cells as they move, as they divide, and see really how these things happen in the end. But basically, this is...
You can find these movies, by the way.
Hey, don't spoil my lecture.
Yeah.
Of course, I show them the video, but this is a virtual practical part. When you look at the microscope, you don't look at videos, right?
Yeah.
So the video is putting together everything and making sense of what we just did. And please don't spoil it. You know, I want to show this video, and maybe you haven't seen it before. Maybe Marcus has shown it in his developmental lecture. But you know, for me, it's nicer if I have the feeling I tell you something you didn't know before. Okay. Okay. I'm actually enjoying this very much. I hope you also have a good time. Do a new share. Okay. Okay. And an hour later, we're at this stage. And now it gets even more obvious. So it's not too much to see. So epibole is done, okay? I give you this much. Okay, this is kind of nice. So this looks like the shield, but it's not. Of course, the shield stage is long past. The embryo, this is called the tail bud stage. The embryo is completely covering the yolk. It looks a bit thicker here than here, but this is kind of strange here. This is this amorphous mass here. I'm going back to the drawings. So here at the bud stage. This is what you see if you would have made a drawing. We already learned that this would be the front and this would be the back. This is where the yolk plug has been, but it's not there anymore. Not much to see, really. This, I can just tell you, this is called the polster. It's not important. This is kind of interesting. This is actually the tissue that will form the tail of the embryo. I will explain that later. But you saw that maybe here in this video here, that this is sort of this thing here. It's difficult to see in the video. Okay. That's that. Any questions? Did you learn something? Not even that. Come on. Shit, what happened? Oh, shit.
It froze.
Yeah. Something, it opened too many. Accidentally opened, okay. Okay. I'll show you a couple of, okay. I won't show you all of these, but the situation is such that now what we just learned is that the embryo is formed. You have basically, you know where all these Anlagen are for the structures that will form, you know, the front of the embryo, the back of the embryo, the body axis is laid down. And now what, of course, cells do is they start differentiating, right? You have, so this basic body plan is laid out, and now the different tissues start forming. And that's what you basically see already before you start. I mean, this is a very nice still to start with. You see that well, I'll show you this video first. See, when he moves it, things get a bit out of focus sometimes, so it's not easy moving and focusing at the same time. Oh, it takes forever. Okay. Basically, I just ask you, what do you see here? Well, this is a head. Just a guess, what could this be?
The eye.
Yeah, that's the forming eye. Do you know the eye, what tissue that is coming from? Is this ectoderm or mesoderm?
I do not know that. Sorry.
Okay, it's actually part of the brain, and it's part of the ectoderm, so the neuroectoderm, and it forms at 13 hours of development. You see, like, it's called the optic placote. Okay. And then the brain is going to go all the way here. This is where the tail is. And now I move a little bit to, let's see. Then you see these things on the side. So that are in 2 rows, there are these blocks. You don't see them here. So that is, yeah, hold on. Here. You know what these things are?
The somite.
These are the somites, yeah. And you know what somites are? What they form later?
And bones and musculature.
Right. Muscles and also the bones of the vertebra. And the dermis. I'm not sure about the dermis in the fish, but mostly muscles, actually. 90% of these cells are becoming muscle cells. Okay. And okay. So this is how the somites are formed. So you see, if you look at this embryo, if you would look, the somites are these blocks of epithelial tissue. And they're forming, so this would be somites in the front. And these are the somites in the back. And actually, what's happening here is a somite that will bud off exactly here from the presomatic mesoderm. So the presomatic mesoderm is here. And then the somites are continuously forming at the front, in the back of the embryo, but in the front of the presomatic mesoderm. Okay. Okay. 8 somite, 20 somite. Let's go to the 20 somite. Oh, this is nice. We go to the 14 somite embryo. So not too many things have changed. You may notice that the brain is more prominent. If I just go back here. Here, see, here, you see where the eyes are forming, and you see much more clearly the structures in the brain already. And what you may also notice in the beginning here, the somites, if you look at the somites, they have changed their shape. Okay. So from a rounded shape like this, they're starting to differentiate into actual muscle tissue, and that is shown here. They become this characteristic, it's a V shape, also called a chevron shape, okay? Like this. And if you look at the cells, they become, you will see muscle fibers contained in these somites. Any questions? No questions, okay. I move directly to the 24-hour video.
Maybe do that with the chorion removal, just to see.
Yes, that's the next one. That's the one after that. It's going to be in the next folder.
No, it's in this folder, I think.
Oh, yeah.
Yeah.
Oh, yeah, that's what it is.
Yeah.
Actually, am I already sharing this?
Yes.
Okay. So here, first thing that you may notice is, oh, you'll see that later better. So this is what you would have done in the course. Your first sort of practical thing that you really would do is, becoming a midwife and giving birth to this fish, getting it out of its shell. And this is very nice to look at. I mean, what is the first thing that you notice? It's so obvious you don't really realize it. This happens with, we notice a lot of things that we're not aware of. And only later you say, oh yeah, okay, of course, I saw that. Oops. See that? Actually, the embryo is moving, okay? This is, in some reason, important. It means the muscles are functioning, but they're not actually innervated at this point. It's not that it responds to any perception input or anything like that. It's cell autonomous twitching, right? It's like micro cramps, if you want. Okay. Okay, but it's starting to move. This has practical consequences for us. So when we do experiments, we actually anesthetize the embryo. Actually, we paralyze them. We paralyze them by putting them into a gel and also put some drugs that prevent stimulation of the muscles under the microscope. Okay.
Also, you see that the shape of the yolk has completely changed. So
yeah. So I'm moving to the next folder because we're approaching 12 o'clock and we have 2 folders to go. And these are really nice to look at, these videos. Okay, and it didn't really work so well this time. So what did we see? Okay, what I wanted to point out still was, which we haven't looked at clearly, was this ear, the inner ear. You will see that later, I guess. And the brain parts. Let's just look at these videos now. So I have too many things open. I have to go to QuickTime Player.
So the 26.
26
Yeah.
blood flow movie, huh?
Yeah,
26?
but then it's only, you know, the full embryo, not the single part.
Okay. Yeah. Okay. I'm a little stuck here for a second.
I have a question about cell autonomous twitching.
Yeah.
Is it due to salts in the liquid, or would this also happen in a distilled?
We have not looked at it. I don't think anybody has really looked at that. It's considered autonomous. I guess I don't think there's real like action potentials or anything like this going on. It's probably well, I don't know what it is. It is not stimulated by motor end plates or anything like this. It's like cells in culture that twitch. I don't think it has whether there's any ion channels involved in that, probably, but it's spontaneous. Yeah. Okay. I can't. Okay, I have to. I'm not sure. So, more questions. I'm still a bit here. Okay. Why don't I? Okay. Let me close a few windows here. There's too many windows open. Okay. Now I got it. You see it?
Yep.
Okay. So it's still moving, same embryo. The structures that we haven't talked about yet at all is the notochord. Notochord is extremely important in development. You will hear about it in neurobiology. I'm very positive about it, that Sylvia Aber will talk about the notochord, or somebody else. It's important for patterning, dorsoventral patterning of the neural crest, of the neural tube, and all that. It's doing the same in fish. So I don't want to go through that. I want to make sure that you see this structure here, which Jamin never focused on, but this is another placode, and it is interesting. It has these two things in there that you cannot see very well here. And you see actually the first differentiating brain parts. You see nice chevron-shaped somites. This is called the yolk extension. And we work on blood vessels, so the rest of the movies will mostly be about blood vessels. But what you usually see if you would draw it properly, you would see these structures here in the picture, and that would be part of the brain. You would see the eye. Has anybody of you done any neurobiology? Developmental neurobiology? Do you know your brain parts? Do you know? So what are the 3 or 4 basic divisions of the brain, of the central nervous system? Starting in front. Let's say this part, what would you call this? Is it the hindbrain? No, it's a forebrain. Okay, so I don't want to torture you. You see different structures here, okay? And And so you've seen the eye, okay? That is, and then here you have the lens. Then you have some kind of division here. There's a line here. Okay. So this is a boundary between the midbrain and the hindbrain. This all is hindbrain. That is shown here in the bottom. This part here, the dorsal part of the hindbrain here, that's the cerebellum. That's important for motor control. The rest here, these are called the rhombomeres. So this is segmented part of the brain. So that's the hindbrain. Same in chick and mammals, 7 rhombomeres. This then here is the midbrain. The dorsal part of the midbrain is called the tectum. And this is where the visual input goes in. That's interesting because when I showed you the video with the paramecia, with these flashlights in the brain, so let's say when the paramecia is swimming on the left side, you would see this calcium release on the right side in the optic tectum here, right? So it was on the other side of the brain. So a left input on the left would lead to a spike in the right tectum, an optic tectum, okay? And this is basically then the differentiation between, in the forebrain, between the telencephalon and the diencephalon. Basically same as, and you will do it, this is neurobiology. So I won't teach you any neurobiology. I don't know much neurobiology, and you will, after the blog course, you will know more neurobiology than I ever did. So this is my rudimentary knowledge, okay? The other thing that is interesting is that you haven't really seen in the videos, and that's this structure here. But it's very prominent if you would look at it yourself under the microscope. What could that be? Just guess. If you're wrong, it doesn't matter. Think about your own head.
The ear?
Yeah. But you have never seen a fish with an ear. There's no ear fish. So what part of that ear could it be? Think about neurobiology. Well, it's not the outer ear. It's not the middle ear. They don't have an outer ear. They don't have a middle ear. They have an inner ear. And actually, it's doing the same as it does in other vertebrates. It's important for balance. It's a balancing organ. So it's a neuroepithelium here. Okay. And then there are hair cells in here. So sensory cells that have little flagellas going out. And these are minerals. This is basically carbon, calcium carbonate here. And these things are actually growing. They are in all fish, and you can age a fish based on the size of these othelids. They're called othelids. They are crystals that form in the inner ear, and they have gravity, and they move when the fish move and when they balance. And that is thought to create a current because it's a vesicle structure, and that current is being picked up by the flagella of the hair cells. And that's the way a cell embryo can balance. So a fish with inner ear problems on one side, let's say on the left side, it will start swimming in circles. If you have a mouse with inner ear problems, it starts running in circles. Because it tries to balance, it cannot balance anymore because there's different inputs on the left and the right side. So it starts running in circles. Okay, give me 15 more minutes and I will show you a couple more videos. And in the end, actually, I show you some more videos of the transgenics because that's really helpful. So, no, let's not do that. I do. Yeah, okay. Are you hungry? So we work on blood vessel formation. So we're interested in how blood vessels are formed, how they interconnect and all that. And what you can already see at this stage is that blood cells are moving here through the trunk. Maybe, I'm not sure. Yeah, you see these little things here moving. You see these specks? Okay. You see it better in a, oops. I have to restart this again.
Maybe if you load on QuickTime one at a time, you won't.
Yeah.
Rather than opening all, you open one at a time and.
Yeah. Okay. So this is the heartbeat. This is now in slow motion.
No, it's not.
It's not in slow motion.
No, you don't see it. You don't share the right window.
Now?
Okay, now yes.
Okay. You start from the front. So in principle, with a high-speed camera, we would be able now to look at, the contractions, and we could look at action potential in the muscle cells of the heart. We can analyze the dynamics of the blood flow. And you see the blood is coming from the flows over the yolk, and it collects under the yolk, and then it's sucked in by the heart and then distributed through the embryo. I stop this for a second. I open a different video. Yeah. Okay. You see it? See an embryo?
Yeah.
Okay. Okay. So just to look at the blood flow, you see also pigment cells. That's no big deal at the moment. But you see there's a lot of blood flow. There's blood flow in the brain. Okay. And I go back here for a second, and then I show maybe one more movie. So at 3 days, you see there's already a lot more blood flow. The heart. You see now on the left side, you see the ear, the inner ear. Okay. Okay. So now I show you a few ways to look at, okay, one thing here that you haven't described are these intersegmental vessels, but you will see them later. I go back to the last folder now to show you some transgenics. For that, first I close this one here. Okay. Actually, okay. Now, there's several ways to look at these embryos. You can just look at them as we just did. Look at them as they come, or you can make transgenic embryos, and you can make transgenic embryos so that express, for example, certain proteins, in this case, DS-RED and EGFP in your tissue or cells of interest. So what we have here is we have an embryo that expresses DS-RED in blood cells and EFP in endothelial cells. These are the cells that make up the blood vessels. And so if you switch now to red, you see only the blood cells, the erythrocytes in this case. So this is under normal light. You can really trace nicely the blood flow through the head. So here the blood is coming from the dorsal part. This is called it's a cardinal vein, the common cardinal vein. And here's the heart, and then you have here the outflow tract. Okay. I'm still struggling here with my sharing here, so I share you for the moment, and let's look at this one. So this is in the trunk. What is important here is there's two main vessels. There's a dorsal aorta, which do you see my arrow? You see my pointer, right?
Yes. Yeah.
Okay. So that's the aorta. So that is the oxygen-rich blood that comes from the heart, and here is the cardinal vein that brings the blood back to the heart. And then, if you look carefully between these somites, you see these little vessels here. You see it's pulsating. And then if you would look for endothelial cells, you see, okay, oops, these blood cells, they move within blood vessels. And then here they are, the blood cells, and you see how they move through these little capillaries. So you see we have big blood vessels, we have small blood vessels, and you see actually that the blood flow is pulsating, and that has to do with the fact, it corresponds basically to the heartbeat. And I give it back to you. And so this is basically it, what we can do in the fish. I mean, we can do a lot more things, but these are the basics in morphogenesis. So we had the embryo axis formation, then we had the first organs formation, and actually the blood, the circulatory system, the cardiovascular system is actually the first organ actually to become functional in embryonic development. That is true in fish, but also in mouse. So if you open up a mouse at 9 days, you have a heart that is pumping blood in the embryo. So that's really a first functional organ in most vertebrate embryos. And I finish with this video here, and that is simply to show that at 5 days, the embryo is fully developed. It's not called embryo anymore. It's a larvae. Criterion of a larva is that is self-feeding. So it starts taking up nutrition from the outside. So for us, that means a larva is considered an animal. And if you do experiments with fish that are older than 5 days, you have to have an animal license. But at 5 days, we put them from a petri dish into an aquarium, and we feed them with paramecia. And then they're considered a fish. They don't look like a fish yet, really, like a zebrafish, because there's also morphogenesis going on later. So there is, at a certain stage, the fish undergo morphogenesis. And then you get the adult form. Okay. So that's it from my side for this morning. I hope you liked it. It was not very tough, I guess. It was tough for me. I had to do all the talking. Are there questions besides what do I get for lunch? No? What time do we meet again?
I think I'll check now the program. I think 1:30.
At 1:30.
Yeah, 1:30, but you can also make it a little later if you want.
Well, I have to give a lecture. No, I can't do it later. We do 1:30. So that's
yeah, and then our group will be meet again at 2:30 or.
Well, we have a break after my lecture.
Yeah,
and then,
exactly, and then I will send again
how lecture
an
goes.
invitation.
Yeah.
For the next thing.
Yeah.
Okay.
So are you okay with this? This is your first virtual block course? Or did you do the microbiology before? Okay. Okay. Yeah. Yeah. Do you think is that okay for you to do this? Yeah. I mean, you have no choice, I guess. Yeah. So the zebrafish part is not part of the exam, okay? But the more you can, take opportunities to learn. I mean, you will benefit from this. You will see there's patterns in different part of this course, right? I mean, zebrafish is like a mouse that swims, and vertebrate development is vertebrate development. So take the opportunity and get the most out of it for yourself. Okay, so I see you in about an hour.
Okay.
Yep. Ciao.
Thank you.
Bye-bye.
Bye-bye. Bye.